ما هو طيب. Now we have to start the first session, so please be seated. يلا ابتدي الأولين بسرعة.
Bismillah ar-Rahman ar-Rahim. Good morning, everybody. We are now starting the first scientific session in the 10th BGICC, and we have many distinguished speakers and many hot topics, and I am honored to be part of sharing this session with my dear friend and colleague, Professor Dr. Ala Andil, Professor of Clinical Oncology, Alexandria University, Professor Dr. Manal Maawat, Head of the Department of Clinical Oncology at Shams University, and Professor Dr. Sami Al-Khatib, our great professor from Jordan. We can allow Dr. Ala and Dr. Manal speaker and one question for everybody?
Yeah, at the end.
At the end? Okay, so the system will be a presentation and we keep the question to the end, please. So now we have a first presentation. Can people on the back of the hall keep quiet and close the door, please? Because this is very high echo here in the hall. So we have a metastatic triple negative. The future begins as well. And this is a very distinguished professor from USA, Dr. Joyce. I cannot pronounce his last name, but she is very famous medical oncology professor. So please come for your presentation.
Thank you very much. Good morning, everyone. Thank you very much for the honor of the invitation to be here for the 10th annual meeting. Thank you very much to Professor Hasham Al-Ghazali for the invitation and to the other meeting organizers. And it's a pleasure to talk to you about triple-negative breast cancer, looking forward to the future. So you may be aware that the Vanderbilt triple-negative breast cancer subclassification has been now simplified in a publication in 2016. Initially, there were 6 subtypes of triple-negative breast cancer, and now there are 4 subtypes of triple-negative breast cancer. There's the basal-like breast cancers that have DNA repair deficiency, high proliferation. Basal 2 has a worst prognosis and has increase in EGFR expression, MET, WNT, IGF-1R. So we have 2 basal-like subtypes. Again, DNA repair deficiency, high proliferation. We have M that stands for mesenchymal, triple-negative breast cancer that is enriched in genes where the cancer changes from an epithelial to a mesenchymal subtype with increased cell motility. And the PI3 kinase pathway inhibitors preclinically appear to be effective against this subtype of breast cancer in preclinical models. And then down at the bottom, we have the luminal androgen receptor positive triple-negative breast cancer, and inhibition of the androgen receptor preclinically is beneficial for this subtype of triple-negative breast cancer. And over here it says, no triple-negative breast cancer subtyping assay is yet of proven clinical utility. Yet this subclassification, I think, helps us begin to organize and try to understand triple-negative breast cancer, at least hypothetically. Now, this Vanderbilt subclassification, I think, it actually dovetails quite well with the potential targets that we have available for therapy. Shown in the red arrows down at the bottom, we have PARP inhibitors, defects in homologous recombination, platinum-based agents, all of which are aimed at homologous recombination deficiency that is seen in triple-negative breast cancer, especially the basal-like breast cancers. The other red arrows point to AKT and EGFR, which may be involved in the more mesenchymal biology of triple-negative breast cancer. And then we also have the androgen receptor that is involved in the luminal androgen receptor positive breast cancers. And so these are some of our key targets now in approaching triple-negative breast cancer. So looking first at DNA repair deficiency and how we might begin to exploit that in triple-negative breast cancer, everyone is well aware of the importance of being able to repair double-strand breaks. And the basal-like breast cancers, many of them have a BRCA-NES, either germline loss of BRCA1 or methylation and inactivation of BRCA1, or other biallelic loss, somatic loss of homologous recombination repair genes. And so many of the triple-negative breast cancers have a BRCA-like phenotype, and this leads to escape and upregulation of double-strand repair via the non-homologous end-joining pathway on the left. And up at the top right, we see that loss of BRCA1 proficiency increases sensitivity to PARP inhibitors with PARP trapping and DNA replication fork collapse. So we have seen already with the recent FDA approval for germline BRCA metastatic breast cancer, we've seen that the PARP inhibitor olaparib, and then also more recently at San Antonio, the PARP inhibitor talazoparib, have high response rates against germline BRCA metastatic patients. And this kind of illustrates the hypothesis of synthetic lethality for triple-negative breast cancers that have lost DNA repair proficiency. They have homologous recombination deficiency, and they benefit from PARP inhibitors. And it turns out that platinum, cisplatin, carboplatin, also form DNA adducts that arrest DNA replication forks and require BRCA1 and 2 for repair. So both platinums and PARP inhibitors both cause replication fork arrest and double-strand breaks. And when the cancers have a failure of homologous recombination proficiency, so they are deficient in homologous recombination, this leads to loss of fork protection, chromosomal instability, and death. So the platinum-based agents and the PARP inhibitors work very similarly against the triple-negative breast cancers that have homologous recombination deficiency. A very important paper was published in the JCO in 2015 by Stephen Isikoff. This is the TBCRC 009 trial. And patients with metastatic triple-negative breast cancer received physician's choice of either cisplatin single agent or carboplatin single agent, either as first or second line therapy. And in the first line setting, the overall response rate with cisplatin was 35%, and with carboplatin was 23%. But what was most interesting about this study is that 6 patients out of the 69 here had multi-year, very highly durable responses. Some of these patients were cured. Now, this only happened first line. This does not happen in the second line setting. It was 11.7% with cisplatin and 5.7% with carboplatin of patients who basically had multi-year survivals and some even cures with no recurrence of disease with cisplatin or carboplatin first line. Now, these patients over here on the left were BRCA wild type. The other thing to note is that the sites of disease were breast, lymph nodes, and lung, not bone and liver. So these patients who had the exceptional responses to first-line platinum therapy were BRCA wild type and had breast, lymph node, or lung metastasis. So please remember that, because I'll come back to that phenotype here in a moment. Now, the checkpoint inhibitors, in this case the PD-L1 inhibitor, atezolizumab, also has been shown to have a proportion of patients with very, very durable responses, as shown here, going out more than a year, even up to 2 years, a subset of triple-negative breast cancers, and they can have durations of response that are approaching 2 years in a subset of these patients. And these patients who have an excellent response, either a partial or complete response to atezolizumab, have very excellent survival compared to patients with stable disease or compared to patients with progressive disease. And at San Antonio, we saw a similar phenomenon in the KEYNOTE study with now pembrolizumab, the anti-PD-1 monoclonal antibody in PD-L1 positive patients, 84 patients, 23% response rate in the first-line metastatic setting. And again, these patients who had partial or complete responses had excellent overall survival. The stable disease patients did fairly well as well, and then patients with progression had a poor outcome. What's interesting about the patients with durable responses to the checkpoint inhibitors is that the sites of durable response to the checkpoint inhibitors are lung, lymph node, and breast. And these sites of metastatic disease has been shown by Shireen Loy to have higher levels of tumor-infiltrating lymphocytes. So we have seen this same phenotype both now in triple-negative breast cancer patients that have exceptional responses to cisplatin, and now in patients who have exceptional responses to the checkpoint inhibitors. Why might we be seeing the same phenotype in both those with homologous recombination deficiency as well as those who respond to immune checkpoint inhibitors? And there's this very important paper that was published in the JNCI in 2017 by Parks and colleague. And what they showed is that in triple-negative breast cancers that have homologous recombination deficiency with double-strand break repair deficiency, that this leads to cytosolic DNA. So the chopped up DNA goes into the cytoplasm, and this then incites an antiviral response in the cytoplasm with upregulation of the STING pathway, leads to the transcription of a 44 gene profile, gene transcription profile, that is called DDRD, DNA damage repair deficiency. And this 44 gene assay had been previously shown by Mulligan in JNCI 2014 to predict for benefit from the DNA damaging agent doxorubicin. But when there's cytosolic DNA because of homologous recombination deficiency and upregulation of the STING pathway, it leads to a type 1 gamma interferon response, and that leads to the production of chemokines such as CXCL10, which then attract into the tumor CD8 and CD4 lymphocytes. So this is an antiviral response that the triple-negative breast cancers mount because of cytosolic DNA. And so this then connects the deficiency in DNA damage repair with increased immunogenicity, because this also leads to increased PD-L1 expression. And what has been shown in this in these preclinical models of triple-negative breast cancer is that the cancers that are positive for DNA damage repair deficiency, so they are deficient in DNA damage repair, they have increased infiltration of CD8 tumor-infiltrating lymphocytes in the tumor and stroma, and they also have upregulation of PD-L1 expression. So we would like to be able to have more triple-negative breast cancers have this phenotype. We want more genomic instability in triple-negative breast cancers that can incite this immune reaction and also predispose the triple-negative breast cancers to cytotoxicity by DNA-damaging agents. And so there's this very nice review in Cancer Discovery called The Enlarging Intersection of DNA Repair Deficiency and Immunotherapy, where increasing genomic instability leads to a better MHC presentation, dendritic cell activation, and a type 1 gamma interferon response. And also, increasing genomic instability leads to the cytosolic DNA, activation of the STING pathway, and a type 1 gamma interferon response. And so these two mechanisms are very closely linked in the triple-negative breast cancers that do well with platinum-based therapy and with checkpoint inhibitor therapy. So we would like more triple-negative breast cancers to have this particular genetic genotype and phenotype. So what other targets can we look at that we might be able to inhibit that potentially could lead to more genetic instability, more DNA damage response deficiency? So we'll look at AKT, EGFR, and the androgen receptor. So it turns out that there's a very good literature in the radiation oncology literature. Radiation therapy in triple-negative breast cancers causes double-strand breaks. And in that context, epidermal growth factor receptor binds to EGFR and then becomes cytoplasmic, and then AKT phosphorylates EGFR, shown here, at its nuclear translocation site, and EGFR becomes nuclear and leads to the phosphorylation of DNA PK. DNA PK and KU70 and 80 are responsible for non-homologous end joining. So EGFR orchestrates non-homologous end joining together with AKT to drive repair of radiation-induced double-strand breaks. And it turns out that AKT1 also enhances non-homologous end joining by stabilizing KU70, one of the key enzymes involved in non-homologous end joining. And in glioblastoma multiforme, inhibiting AKT1 increases sensitivity to etoposide. So this then points to AKT and to EGFR as mediators of non-homologous end joining, which can decrease double-strand breaks and decrease sensitivity to DNA-damaging agents. We have seen early data from the LOTUS trial, randomized phase 2 trial, in first-line patients with metastatic triple-negative breast cancer, those with a PIK3CA mutation, AKT mutation or amplification, or loss of PTEN genomically. This subgroup of patients had benefit from ipatasertib, which is an oral AKT inhibitor, paclitaxel alone, or paclitaxel with the AKT inhibitor ipatasertib, had an improvement in median progression-free survival in triple-negative breast cancer patients, which is quite encouraging. Also, we have seen from the I-SPY2 clinical trial with the Merck 2206, another oral AKT inhibitor, where patients were randomized to weekly paclitaxel followed by doxorubicin cyclophosphamide, or the AKT inhibitor with weekly paclitaxel followed by AC. And in the triple-negative population, there was nearly a doubling of pathologic complete response with the AKT inhibitor. And this did graduate, but this will not be pursued in further trials in the Merck compound. But the ipatasertib has completed a randomized phase 2 called the Fairlane trial, preoperative paclitaxel with ipatasertib or placebo, followed by surgery, then followed by AC, and we await the results of this clinical trial to see if ipatasertib, followed by DNA-damaging agents, will potentially increase the sensitivity of the patients to AC chemotherapy by inhibiting AKT. EGFR also regulates non-homologous end joining because it regulates the production of this long non-coding RNA called LNP1, and this LNP1 is increased transcription because of EGFR, and this long non-coding RNA then acts as a scaffold protein to bring DNA PK together with Ku70 and 80 to drive non-homologous end joining. So the EGFR is very involved by multiple mechanisms in non-homologous end joining, which would then decrease the sensitivity of triple-negative breast cancers to DNA-damaging agents and potentially as well to checkpoint inhibitors. There was a very important paper published in 2012 in Cell showing in triple-negative breast cancer preclinical models, cell lines on the left and tumor xenografts on the right, that if you give doxorubicin alone or the EGFR inhibitor erlotinib alone, the percentage of apoptotic cells is about 15%. If you give the doxorubicin and erlotinib together, you don't get any increase in apoptosis. However, if you give the EGFR inhibitor before the doxorubicin, you get a 500-fold increased sensitivity to the DNA-damaging agent doxorubicin. So giving EGFR inhibition first and then coming in afterwards with the DNA-damaging agent greatly increased cell kill. And shown here in red, we see that during the erlotinib, the cells are growing in xenografts during the erlotinib, and then you come in with the arrow with the doxorubicin, and you see very substantial cell death with the doxorubicin. And if you give the erlotinib and doxorubicin together in the blue, you see good cell death, but then the cells start to escape. But with the erlotinib first, followed by the doxorubicin, you see a prolonged cell kill. And do we have any clinical data to suggest that this might actually help patients? And we saw from the I-SPY-1 trial in triple-negative breast cancers that there was a small group of triple-negative breast cancers that had phosphorylation of HER2 and EGFR. So these were not amplified for HER2. They were triple-negative breast cancers, but they were co-phosphorylated with EGFR and HER2. And we have also seen that in doing PAM50 intrinsic subtyping analysis of triple-negative breast cancers, that 17% of them are HER2 enriched. And this is the same group. This is a small subset of triple-negative breast cancers that are phosphorylated at HER1 and HER2. And in the I-SPY-2 trial that looked at preoperative neratinib, and neratinib is a very powerful inhibitor of EGFR and HER2. The neratinib did not increase the pathologic complete response rate in triple-negative breast cancer, but in the 40% of the cancers that had phosphorylation of HER1 or phosphorylation of HER2, the neratinib did increase the path-CR rate by about 20%. So again, inhibiting HER1 and HER2 before treatment with AC chemotherapy was beneficial to patients in terms of increasing the path-CR rate. It turns out as well that the androgen receptor leads to the transcription of a whole host of DNA repair enzymes, and in castrate-resistant prostate cancer, enzalutamide suppresses expression of DNA repair proteins and creates BRCA-ness and sensitizes the cells to subsequent treatment by olaparib. So I will stop here and just finish up by saying that you can prime triple-negative breast cancers to increase homologous recombination deficiency and immunogenicity. A wide variety of agents can decrease DNA repair proficiency and increase sensitivity to DNA-damaging agents, and there are a number of agents being looked at in this regard, and that is true as well in inhibiting AKT, inhibiting EGFR, and potentially in inhibiting the androgen receptor. Thank you very much.